Hello learners, welcome once again to the last part of our ongoing series on the mechanism of photosynthesis. Now naturally this is entitled as what is the rationale for the C4 and the CAM cycles. Remember learners, in the last lecture we had asked ourselves a few questions regarding the evolution or simply the need for the origin of alternative carbon dioxide concentrating mechanisms in photosynthesis. So, talking about the C4 cycle that we had mentioned in the last lecture, we found that there were two cycles in fact running together, and they were running simultaneously but in different compartments. So, this was a case of spatial isolation. Spatial isolation, they were occurring at the same time, but in different compartments. And talking about the energy input of the C4 cycle, we had also reached a conclusion that C4 cycle was more energy consuming and it was consuming 12 ATP more than the C3 cycle. Yet, this high energy input was overcompensating the energy losses that these plants would have incurred in the presence of high oxygen were they C3 plants. Now, we ask ourselves a question, and that is, if the Hatch and Slack pathway helps to prevent photorespiration, then the question is, why won't all the plants have this adaptation? Why are we then talking about C3 plants in the first place to be universal? Now, the answer is very simple, that is, the answer is not in our hands, the choice is not of a plant, it is the mercy of the species evolutionary history. How this particular species has evolved in nature, what were the set of environmental conditions prevailing at that time, what was the combination of the selection pressures which were present in the ancestors, and whether these ancestors had a mutation, and whether this mutation actually caused the adaptation which turned out to be favorable for them fortunately. So, that means all these questions have to be answered in terms of the evolutionary history. We know that C4 cycle is present only in angiosperms. So, which gives us a very important food for thought, and that is that this evidence would suggest that it would have a multiple independent evolutionary origin. It is not coming from the lower plants as such, and the C4 pathways perhaps evolved themselves in response to low CO2 levels and the need emerging from it therefore to concentrate it further and to supply it to rubisco. And we find that aridity, that is the arid conditions, seem to be a very important factor. Let us look at some of the examples of the plants that have Kranz anatomy, and they are the C4 plants, and naturally they are more efficient than the C3 plants. They may belong to the family Acanthaceae, Asteraceae, that is Compositae, Caryophyllaceae, Cleomaceae, Nyctaginaceae and Scrofulariaceae. In addition, we find a very interesting situation. There are certain genera belonging to the family Isoaceae, Amaranthaceae, Asteraceae, Chenopodiaceae, Euphorbiaceae, Nyctaginaceae and Poaceae, which is Gramineae, which have representatives of both C3 as well as C4 species. Now, this is a very intriguing and a mixed situation. If we took an example, looking at your screens, the anatomy of two species of Atriplex. Atriplex petula is a C3 plant, and you know the anatomy of a C3 leaf. But the Atriplex rosea, which is growing under more dry and arid conditions, has a typical Kranz anatomy. So, that means two species of the same genus growing under different environmental conditions in nature may evolve different internal structures and different physiological attitudes or the processes. Now, even a hybrid was made between the C3 and the C4 plants, and the hybrid in fact showed intermediate anatomy, and in the F2 generation it showed mixed C4 and C3. Now, we are not employing Mendelian principles here. The basic idea is evolutionary. What do we conclude? We conclude from this whole hybridization experiment is that the enzymes which are specific for C4 photosynthesis are already present in the C3 plants. Whether they are given a chance to express themselves or to function is a different matter altogether. And this different matter is of course controlled by the environment. So, that means it also gives us an idea that C4 photosynthesis perhaps evolved from the C3 metabolism by changing the specificity and the regulatory properties of tissues and the specificity of key enzymes. And it also led to a physical separation of carbon fixation from the Calvin cycle. As you know, learners, there are not just one, but various types, different types of C4 cycles, which are taking into account how the carbon dioxide which is released into the bundle sheath cells is variable, and therefore, we have different types of CO2 concentrating mechanisms which are present, which is not in the purview of our discussion today. What we have a very interesting aspect is that the C4 photosynthesis, as we said earlier, belongs only to the higher plants. No, it has now been discovered that there are certain unicellular protists, which are the diatoms in algae. They also show certain membrane bounded bicarbonate pumps. They are going to trap bicarbonate and then they are going to transport it to Rubisco rich carboxysomes. So, that means single celled C4 photosynthesis is also possible, and that too without Kranz anatomy. So, in other words, we say that the Kranz anatomy may be very important and a prerequisite for the desert plants, but then the individual chlorenchyma cells would have further specializations, and the single celled organisms would be all in one. So, that means there the chloroplast itself would be partitioned and not the two types of chloroplast as we saw in terms of dimorphic plastids. There will be a central compartment and a peripheral chloroplast compartment, and these two compartments will isolate the two carbon fixations, namely the C4 cycle, that is Hatch and Slack, and the benzyl Calvin, and therefore, the Kranz anatomy would not be needed. So, that means very, very specialized protein rich micro compartments which contain enzymes for photosynthesis are called as carboxysomes, which are important for single cell organisms Hatch and Slack cycle. If you were to see some of the examples of the unfortunate ones. Why we call it as unfortunate ones? Because we saw in our earlier lectures that the C3 plants are nearly 40% less efficient productivity wise as compared to the C4 plants. So, we also said that grasses, they show C4 cycle, but here are some notable exceptions. Wheat and rice are monocots, no doubt, yet they are C3, and therefore they have less productivity than maize and sorghum. What is important here is they are monocots, all right, but then they are not growing in tropical climates. They are growing in temperate climates. So, that means their adaptation, in spite of being monocots, is that of a C3 plant. Learners, let us now switch over to another very interesting aspect, and that is the crassulation acid metabolism. Let us go to conditions of environment where the climate is even drier and even hotter. So, that means more and more stress for photosynthesis, and water availability is only seasonal. For example, succulents, cacti growing in some of the hottest deserts of the world, and epiphytes, members of Crassulaceae, members of Bromeliaceae, Cyperaceae, these grasses, they are all very, very dry, and in a desert the water is the limiting factor. So, these are the common desert plants, the cacti and the succulents. The example also includes certain food crops, and that is the fruit trees like the pineapple, and of course, you can see on the screens the cacti of the Arizona desert and the succulents thereof. Now, in this special type of a cycle, reactions are going to take place during day and during night. Now, I never said that they are going to take simultaneously. So, that means here the reactions are going to show a temporal variation. A spatial variation is all right and in different tissues, but then temporal means occurring at different time periods. Surprisingly, this crassulation acid metabolism also occurs in gymnosperms. For example, in members of the family Welwitschiaceae, which is in nettles. So, that means it also proves that when it came from gymnosperms also, that it must have originated earlier than the C4 plants. Even surprisingly, certain pteridophytes like members of the family Polypodiaceae, Isotaceae and Vitariaceae, they also show CAM cycle. And CAM cycle, the mention was made as early as in 1804, when Nicolas de Saussure, the French scientist, observed that in Opuntia, the illuminated plants could produce oxygen even in the absence of carbon dioxide. And there goes a very interesting story. An Englishman, Benjamin Hayne, who was stationed in India in 1812 and 1813, made some very interesting observations on bryophyllum. Learners, you know, bryophyllum is a plant which we often teach when we are talking about the vegetative propagation of the leaf plantlets coming from the notches at the edge of the leaves. Now, this is a succulent plant. He found that in bryophyllum, which was growing in his garden, the normal taste of herbs in the afternoon was different from when it was chewed in the morning. In the morning, it was very, very acidic. It could even burn the mouth and the tongue. But then towards evening, it was more tolerable. So, it was a very interesting observation, and Hayne even reported these observations through a letter to the Linnean Society in as early as 1813, but then no cognizance was taken of. A CAM plant, that is Crassula, was the member in which the cycle was discovered, and also in bryophyllum, and both of them, they belong to the family Crassulaceae, and hence the name crassulation acid metabolism. Now, why this metabolism has come, that is the rationale, because such plants are growing in extremely dry and hot habitats. So, that means the conditions are very, very stressful for photosynthesis. These are the cacti, succulents, as I said before, even many orchids. So, under desert conditions, what is going to happen is these plants cannot afford to let their stomachs open during daytime, otherwise they would transpire to death. So, during the day their stomachs are closed and they open only during night. You remember, learners, this is a case of scotoactive opening as against the normal photoactive opening. So, that means high concentration of CO2 would be there in the leaves because the stomachs, they are closed, and C3 and C4 cycles are occurring here also, but then they are occurring spatially, that is in different parts of the same cell, as well as temporally. So, what happens during daytime? C3 cycle occurs and C4 cycle occurs during night. Now, this is a very interesting story of compartmentalization, because how can photosynthesis take place with closed stomachs is an interesting aspect. So, what happens is that at night time, when these stomachs are open, there is a fluctuation which we call as reciprocal diurnal fluctuation of malic acid and the stored sugars or glucans. Now, there is a temporal separation of carbon dioxide capture and rubisco. So, what happens is C4 plants, as we saw, are only spatially isolated, but here the temporal one contribution is also there. So, what happens is, to avoid transpiration and photorespiration, stomachs will open during night and they will be closed during day. So, when night comes, then C4 cycle operates and C3 cycle will operate during the daytime. CO2 fixation at night, which we call as nocturnal CO2 fixation, how is it going to be possible? The PEP or the phosphoenol pyruvate is the primary acceptor, which is a 3 carbon compound. The enzyme is pepcase, that is PEP carboxylase, and this is called as dark carbon fixation. It is the same pattern as we find in C4 plants. Now, Krebs cycle is also going on during night, because respiration goes throughout day and night, all 24 hours. So, the CO2 which is released is also now obtained endogenously. The stomachs are open. So, the CO2 availability is there. There is a high concentration of CO2 which will not be toxic, because the moment CO2 is got and even CO2 is taken from the atmosphere, it is very, very rapidly utilized for the synthesis of malate by the enzyme PEP carboxylase. You remember, learners, that PEP carboxylase is many, many times more efficient than rubisco. So, the immediate product OAA is formed, it forms malate, and malate now goes on accumulating. There is a pumping of malate at the expense of energy into the vacuoles. So, the vacuoles are full of malic acid. If you were to now chew the leaves of a succulent plant during early morning, you may even burn your tongue and mouth, because it will be so highly acidic. pH would be ranging, say, around 3. And therefore, there is no wastage of CO2, it is being concentrated throughout night and in the form of accumulation of organic acids into the vacuole, and this is a case of acidification of the cell sap at night. What is going to happen now? The day comes, sun rises, temperature becomes unbearable, sunlight becomes very, very problematic, and the stomachs, they get closed. When these stomachs get closed, then these very acids which were formed during night will gradually get decarboxylated during daytime, and the carbohydrates are formed. So, that means C3 cycle would be obtained, and the carbon dioxide which is so produced in the process and also by respiration is fixed by the Calvin cycle. So, that means if we were to look at a diagram on your screens, you find that both these processes are occurring at different times and in different compartments. So, we call this as a spatial as well as temporal isolation. On your left on the screens, you will find that when in dark these stomats are open, then we find that atmospheric CO2 finds its way inside, respiratory CO2 is not lost, immediately it is converted into HCO3 by carbonic anhydrase, and PEP combines, gives rise to OAA and malic acid. The malic acid goes and gets stored in the vacuoles. During day, these stomats are closed, and we find that this malic acid from the vacuoles now is channelized to the chloroplast, where it gets converted into pyruvate, and ultimately C3 cycle prevails to give rise to starch. So, these plants are unique, and you will find that CAM plants are present in areas where both C3 as well as C4 plants, they will fail to survive, but the CAM plants would be able to be present there. So, that means it is this very ability to close these stomats during night time which is going to help these plants to prevent loss of vital carbon during severe drought conditions. Now, interestingly, some of the CAM plants also exhibit a facultative approach in physiology. They can shift their photosynthetic mechanism in response to variations in environmental conditions. For example, there is an ice plant, Misembryanthymmum crystallinum. It belongs to the family Isoaceae. Now, this particular plant, even agave, is a typical CAM plant, but if you give abundant water, so in some years you will find that there is a situation of abundant water supply, it may even switch over to a C3 mode, because now the conditions are much more favorable. Another interesting aspect comes that a member of the family Bromeliaceae, which is Tillandsia species, is not a succulent. It does not store water, yet it photosynthesizes by the CAM cycle. So, that means it also must be present in conditions which are of severe drought. As far as the efficiency of this type of metabolism is concerned, we are sure that CAM is not as efficient as C3 or C4, but you should appreciate that this is the best suited for those conditions which are extremely water deficient. So, from the point of view of evolution, it is believed that the occurrence of CAM cycle in primitive ferns, even in Welwitschia, it is present. It also indicates that this is a mechanism, or this mode of photosynthesis is to be it was evolved earlier than the C4 mode. So, that means it must have originated in the past 35 million years to capture atmospheric carbon dioxide and even scavenge the CO2 conditions. So, to conclude, learners, we have on your screens a comparative pictorial account of C3 photosynthesis, C4 photosynthesis and the CAM cycle with the examples of rice, maize and pineapple respectively. I would again repeat that these three cycles are present in plants not by their own choice, but by the evolutionary mechanism and the selection pressures of the environmental conditions which have prevailed, which have helped them to evolve in these different directions. Thank you.